Hi, I'm Dr. Matthew Sharps, and welcome back to the world of the mind. In our last episode, we talked a bit about memory and how it works. Now, memory is fantastically mysterious. It really is, but it's also fabulously important. Memory is pretty much what we are, if we think about it. If you're an architect, you probably want to remember that you're an architect and don't decide suddenly that you're a dentist or something. And also, if you don't want the buildings to fall down, it's a good bet that you remember something about architecture, all right? So memory is critically important, and we'd really like to know more than just how it works, as we discussed in our last episode. We're going to be discussing that more in future episodes too, but for the moment, what we're going to ask is a very difficult question. What exactly is a memory? What is one? Now, that's an important question. Are memories real? Well, yes, they obviously are real because they obviously have effects in the world. If your architect and your dentist don't remember what they're doing, your teeth will fall out and the building will fall down. So you've pretty much got to have memory work, all right? It has effects in the world. It's a real thing. But what kind of thing is it? Now, that comes to a concept called the engram. That's E-N-G-R-A-M. That's the physical basis of memory. And we'd like to know what a memory is. Now, does a memory exist the way, for example, this pencil exists? Well, it has to some degree. If you think about it, it has to have characteristics. Now, take this pencil for a second. First of all, it's yellow. I can see that. Secondly, I know that it's made out of wood. It's graphite in it. And since I do know what a pencil is for, I can write with it. And this particular one has a little bit of rubber here. We can use it as an eraser, okay? And it's got this metal here holding the eraser in place. Now, I don't personally know what kind of metal it is, but I don't need to. It doesn't need to be in my memory. Now, if I were a pencil manufacturer, I would damn well want that in my memory. It'd be kind of a problem if I didn't. But since I'm just going to use this pencil as a pencil, that's not really important. I can just realize, okay, the eraser is held in place by a piece of metal. Okay, great. So we've got a pretty good handle on what this pencil is and even what we could use it for. We could also use it maybe to poke little holes or something. We use it to move stuff around, but it's basically a pencil, obviously. The thing is, we also know this pencil is not suddenly going to be usable as a pipe wrench. We also know that it's not going to suddenly join a pack of hyenas on the savannas and go chasing after various prey animals. This is a pencil, not a hyena, and it's got that going for it. Now, the problem is, memory doesn't work that way. So we've already seen memory changes into other things. Memory changes, the work of Bartlett, right, from our last video. In terms of just brevity and personal belief, memories get shorter, but they also change in the direction of how we think they ought to have gone. And that's a huge problem for the eyewitness memory. That, again, will be a source of a number of our future videos. So if this pencil suddenly turned into a hyena or an iguana or something, it would be time to talk. But memories do that all the time. So their existence, what exactly is a memory, is a crucial question. I may as well give you the punchline right now. We're going to do this video, but we're, at the end of it, not going to know exactly what a memory is. So the obvious question is, why in the world would you waste your time on it? Well, in previous videos, I've talked a bit about Plato's dialogues, these wonderful dialogues, and a number of these dialogues in these wonderful books here are what we call aporetic. They don't lead to a final conclusion. Okay. One of the dialogues in this particular book by Plato, the Meno, right? Well, Socrates is going on and on with a guy named Meno, and they're talking about what is the nature of virtue, and Meno says, well, virtue is getting gold and silver. Well, that works, but Socrates says, no, couldn't you get gold and silver in a non-virtuous way? And Meno says, well, that's possibly true. Yeah, I got to admit that. So, but maybe virtue is something else, and Socrates says, no, there's these problems with that. And Meno says, well, fine, what do you think, Socrates? What is virtue? And Socrates says, well, it's this, and this, and Meno's going, oh, that's a pretty good explanation. And Socrates basically goes, but there's this problem, this one, right? It turns out that's not it either. At that point, basically, Meno starts accusing Socrates of mouthing off, and Meno's buddy Anytus gets ready to feed him the hemlock. Okay, so the whole thing doesn't work out very well. So why in the world would you want these aporetic dialogues or an aporetic talk like this one? We're not going to get to the final conclusion, really. Well, it goes back to what I like what Professor Michael Segru talked about. The domain of ignorance versus the domain of knowledge. What do we know and what don't we know? And sometimes it's as important to know what we don't know as it is to know what we actually do, because that defines the domain of our ignorance. What do we need to know, and possibly, how could we best start to look for it? So that's why we really want to consider this search for the engram, for the physical basis of memory, because what exactly is a memory? where does it live? Well, ancient peoples, as we've seen, used to think that the soul was in the heart, because if you poked a big knife into the heart, the person pretty much stopped breathing and that was it. Okay, so it makes some sense. We now are pretty certain that's not the case. Okay, the mind, if you want to call it the soul, whatever you want, the mind is basically operating in the brain. We've known that since work several hundred years ago by a group in England called the Oxford Circle. The brain is the seat of the mind. So it's a very good bet that the brain is the seat of memory. And there's a specific structure inside the brain, in the limbic system, okay, the old mammalian brain. It's called the hippocampus, which is a wonderful word, okay. If you speak both a bit of Greek and Latin, hippocampus, it sounds like you're saying horse field, okay. That's not correct, okay. It's straight Greek, it means seahorse. Because the two bits of the hippocampus kind of curve through the inside of the brain, right about here, I guess, right down about here, in the way you might see a couple of seahorses swimming along. So the hippocampus, we used to say, in fact we still do often, that it's the organ of consolidation. That's the place where memories go from this relatively dynamic state of short-term memory to the more permanent, if still changeable, state of long-term memory. Okay? Now that's a lovely concept. But modern neuroscientists are starting to go, that's not quite accurate. They're starting to refer to the hippocampus less as the organ of consolidation than as the organ of encoding, where material first gets into your brain. And that makes a certain amount of sense. Now, there's no actual delimiter, like we've got one circuit going to the next, where we change encoding into consolidation, okay? But we now know, there's a lot of good neuroscience backing this up, that consolidation goes on way beyond the old 15 to 30 seconds that used to be the idea of short-term memory. It goes on for days, possibly weeks, possibly even months, all through the cortex. If we look at this lovely model brain here, it's the right size, right shape, almost the right color for an actual human brain. The cortex is this big wrinkly bit on the outside. And basically the hippocampus seems to send signals up there, and additional consolidation happens in all sorts of locations here in the cortex. So consolidation is a much more complex process than we used to think it was. But we are pretty sure, okay, fair enough, we know where memory lives. It lives in the brain. But what exactly is it? Because it doesn't have the same existence of this pencil. This pencil can't really turn itself into a pipe wrench or a hyena. But memories change. So there's obviously a dynamic quality to memory that we're going to have to deal with as memories move through time. And we're going to be talking more about that in future videos. Right now, though, we're concerned with what we need to know if we want to understand the nature of the engram. As you know, the world of the mind comes in three basic streams here. We have the mind, and then ideas about the mind, effectively the history of those ideas, and then the mind at the extremes. Well, this particular video is both the mind and the history of it, because we can't get away from the ideas, from what's been thought about the nature of memory. So if we're going to have a memory, what exactly is it that we've got to have? What do we need to actually exist? Well, the answer is really two things. First of all, unless memory is running around the immortal soul in some supernatural sense, which we really doubt quite firmly, memory has to involve a physical change in matter. There's got to be some kind of change in matter, and we presume that's in the brain. But that's not enough, okay? If a rock falls on a dinosaur 80 million years ago, well, maybe the dinosaur remembers it, but no human was around to remember this, okay? If there's a rock falling all by itself, that's a physical change in matter, but it's not a memory. To have something that you remember, to have a memory, you have to have two characteristics. One is a physical change in matter, and the other is that it has to be read. Now, I can illustrate this with this lovely piece of blank paper here. Suppose I make a physical change in matter on this piece of paper. I use this magic marker to write a lovely A on my piece of paper. And now we have a nice rude analog, really, of a memory. We've got a physical change in matter, and if you're listening to this talk, you do understand the International Standard Latin alphabet. You know that's an A, so we've got that going for us. We've got a physical change in matter, which is read. So we're looking for something like this in the brain, all right? But where and under what circumstances? Well, an awful lot of what we learn about the brain, we learn from, frankly, lesions, which is a lovely neuropsychological and neurological word for holes in the brains, lesions. And one of the best places to get lesions is in a war where people get shot in the head. Now, through the 19th century, you often seem to see this tacit concept that somehow or other memories are actually in large physical structures of the brain. So, for example, maybe this wrinkle here could have Christmas of 1894, and this wrinkle up here could have Easter of 1897 or whatever. You get the general idea, okay? That is conceivable, but you really have to believe this only in terms, oddly enough, of a military technology. Now, what am I talking about here? Well, people in the 19th century, when they get shot in the head, got shot in the head of the weapons of that day. And in the early 19th century, those weapons fired simply enormous bullets. Now, I've got one here. This is the kind of ball, the kind of bullet, that would have smashed you in the side of the head in the War of 1812, or if you were a fur trapper in the American West in the 1820s, something like that. You compare this mighty bullet here to this brain, you can see it's going to do a hell of a lot of damage. And these bullets flew rather slowly, which sounds good to start with until you realize, my God, that means it's going to go in there and start twisting and turning around, doing tremendous amounts of damage. And the fact is that if you were smacked in the head by a 69, even a 50 caliber ball, 69, it's over 2/3 of an inch across. That's an enormous damn thing to have smashed into your head. Generally, you died. Now, dead people are remarkably bad at reporting their memories. So there's a very good bet that just being dead, well, fair enough. So you get shot in the head. It probably wiped out all the file cabinets from 1812 to 1830, and then you died. And, well, okay, so fair enough. You could believe in these file cabinets in the head. Each wrinkle is kind of a file cabinet. Maybe it holds the year 1817 or something for you. You could believe it. But bullets changed. Now, look at this. This is a cartridge from a .22. And the whole thing, by the way, doesn't fly into your head, just this little gray bit. But look at the difference in the size here. That's only 1/5 of an inch across. And it travels at much higher rates of speed. And this .22 is apparently the most likely thing to be shot with in the modern world, the modern civilian world. Wait, we're talking about military things. Yeah. But the modern M16 is basically a .22, 5.56 millimeter, that's its caliber, if you will, is about the same diameter. It's a different shaped bullet. There's a lot more powder behind it, but it's basically a .22. And other military rifles are larger, about 1/3 of an inch across, the .308, the old 30-06, right? That was almost 1/3 of an inch across, 30% of an inch, invented in 1906. Well, the point is, that's still a relatively small bullet compared to this monster, and it goes very, very fast. And so at the end of the day, you've got this interesting thing that as of the 20th century, really, as the 20th century develops in all its blood-soaked grandeur, people get shot in the head and survive. And it turns out that you don't ever really lose Christmas of 1895 or, let's go into the 20th century, Easter of 1917. Now, what you do tend to do in the amnesias, the pathological forgettings, is forget a chunk of time. And you can lose a chunk of time in the future. You can not be able to learn new stuff. That's an anterograde amnesia. And you can fail to remember the past. That's a retrograde amnesia. And generally, it's not always the case, but generally, the more severe the trauma, the more you basically forget. So if you smack your head on the wrestling mat and go unconscious in phys ed, you might lose 2 or 3 minutes. You smack your head on a moving train, you might lose several months. You get the general idea here, okay? But you don't wind up losing a specific memory. Now, nothing is very clear in this field. Okay? There is a sort of an exception to that. That's focal retrograde amnesia, where a person forgets a chunk of time. And often that's not a physical trauma. Often that's a psychological trauma. People will forget the couple of weeks they were in the field in the military before they whacked by artillery caused this amnesia, or they might forget the period of their childhood where they were molested by a relative or something. Okay? But that's a different thing. We're going to devote an entire video later in this series to focal retrograde amnesia. It's a very strange concept. But the point is, you don't tend to forget an event. You might forget a whole bunch of events over a period of time. But the idea of suddenly we put a bullet through and we knocked out Christmas of 1974, that really doesn't happen. Okay? Now, there's ordinary forgetting all the time. You might forget Christmas of 1974. What did you get for Christmas? Where were you? You might forget that. But it's not a pathological forgetting. It's not caused by putting the zap on the head with a bullet. All right? So suddenly we've got this interesting problem here. By the middle of the 20th century, it's very clear that you don't have a specific wrinkle of the brain containing a specific memory. That idea becomes completely outdated. So, well, where could memory be then? Well, by then, we've started to isolate the cells of the brain, the neurons and the glia. Now, the glia are basically support cells. Nobody has seriously suggested they're involved in memory. There's a few fringe ideas, but nothing that holds water. But the neurons, the actual cells that carry the circuitry of the brain, that carry the messages, that would make sense. Because you could have what we would call reverberating circuits. In other words, the information, whatever it is, would enter the neurons, and the neurons, which are interconnected through their axons and dendrites, the messages come into a neuron through the dendrites, they go out through the axon, right? And an axon, in brain neurons, they make tremendous numbers of connections, all right? So you have all these wonderful circuits there. Well, so you could learn some Christmas that grandma's cookies taste like pure evil, all right? And so that memory of, for God's sake, don't eat grandma's cookies, they taste like pure evil, was circling around these wonderful reverberating circuits. They're reverberating around the brain. And then, when next Christmas comes, you go to grandma's house, she passes you this cookies with flavor of iguana or whatever it is. You realize, okay, dear God, do not eat those cookies. Okay, so it makes a certain amount of sense, doesn't it? Reverberating circuits. We know the brain cells are there. And the bullet missed those brain cells. Maybe those brain cells are talking to brain cells over here. So we wipe out these wrinkles of the brain, these brain cells over here, take it over. They reverberate the circuits, and you realize you don't eat the damn reptile-flavored cookies. Okay, that works. But is it true? Well, there was some wonderful research on this concept by a great scientist named Kandel. He won the Nobel Prize for this. It's really brilliant research. Because there's a kind of marine snail. It doesn't have a shell, and they range from about this big to about this big. I've seen them actually this big here on the Pacific coast. They basically look kind of like a really ugly, dull purple football with some stuff coming out the back. That's their gills and their gill mantle. Now, I've got to admit, I'm not crazy about this as a model for human cognition, because the brain of this thing is actually down where its stomach is, basically. It's not a very good model of mammal or human cognition. But it does have the same sorts of neurons we do, except in aplysia, this wonderful snail, this aplysia. And you get a lot of them live even today are in the central coast of California. The Elkhorn Slough is where a lot of them are taken out of here in central California. Well, these snails have a very interesting property. Their neurons are enormous. And so you can get various kinds of probes in there and read what the neurons are doing. Now, you probably are aware that neurons, inside the neuron, there's an electroionic factor as the ions change inside the neuron. But between neurons, there is chemical communication. And the chemicals are, of course, referred to as neurotransmitters. There's a hell of a lot of them. It's very difficult to know exactly what a neurotransmitter is. The technology is very complex. There's at least 70-odd neurotransmitters at the moment. More turn up all the time. But the point is, the neurotransmitters go from one neuron to the next neuron, allowing the circuit to be completed. Now, this means that if you could read these circuits when the animal has had an experience, you might be getting at a memory. And that's what Kandel did. Because if you stimulate the gill mantle, that's the part that guards the gills of this snail from being eaten by fish, okay? Well, there's parts of it you can stimulate that it habituates. It slows down and eventually stops working. The other parts that sensitize, you keep zapping it and it gets bigger and stronger responses. Well, when Kandel studied this, giving these animals these experiences, and this was done by a number of laboratories, both in sensitization and habituation, what we find is that we zap the poor snail, it's an experience, and we find that it puts out more or less neurotransmitter depending where we zap it. All right? Now, the putting out of neurotransmitter is a physical change in matter. The nerve cell is doing something. It's a physical change in matter. And this cell is putting out more transmitter if it's sensitizing or less if it's habituating. Okay? And the next neuron is reading that. We've got the basis of memory. We've got the idea of a change in matter that's being read. Okay? And it's a wonderful concept. And it's one of the reasons I think that Kandel received the Nobel Prize. I'm not entirely clear on exactly how that's done. But okay, this was a huge deal. We finally got something that philosophically works. Reverberating circuits could be the nature of memory because you've got a physical change in matter, the amount of neurotransmitter that's put out there, based on this experience, and it's being read by the next neuron down the line. So it works beautifully. The only problem is this, all right? Neurons are electrochemical organs, all right? I shouldn't say organs, really. They're electrochemical cells, right? It's a chemical transmission between the cells, the neurotransmitters, but what's going on inside the neuron is electroionic. And that means they can be recorded by electrical devices, okay? Well, that means it's subjectable to electronic interference. Now, suppose you have a person who has an epileptic seizure. Well, this is a result of, well, you might, and this, I can see neurologists getting angry about this if I say this, but you have sort of a storm, really. You have a lot more electroionic activity happening in the nervous system at a certain place, sometimes in the temporal lobes, and it basically causes a hypersynchronization of the brain that creates the seizure. Now, that means that the neurons are going to be subjected to this massive sweep of effectively electrical energy across the brain. And electroconvulsive therapy has come back as there's less and less money to manage patients. You need more management tools, and electroconvulsive therapy is one that's often used with very, very depressed patients. You basically step down the household current and you blast it across the frontal lobes, and that's a bit oversimplified, but you get the idea. The point is you're putting a hell of a lot of electricity across the brain. Now, so what? Well, an epileptic seizure puts electricity across the brain. And electroconvulsive therapy puts a lot of electricity across the brain. Now that ought to, therefore, wipe out any memories that are operating in reverberating circuits. The electrical system should cause the electroionic system to completely blow up. But it doesn't happen. Now what does happen in a very strange way is you will produce amnesias. But they're relatively brief. They may go back a couple of minutes, 2 or 3 minutes. I remember one patient I dealt with many, many years ago who'd had a number of electroconvulsive shocks, and he had actually lost several months. But the point is, most of his memory was still there. He knew who he was. He knew what his occupation was. He was still able to do his occupation. If he'd been an architect, he'd know he was an architect. He'd know how to do architecture. Okay, but so you get the idea here. The short-term memory seems to be blasted out by a big electric shock across the brain. On the other hand, you can do the same thing by whacking somebody in the head with a frying pan. Okay, so we're not quite clear on the dynamics there. What we do know is long-term memories don't go bye-bye. So you've got this huge problem, really. Either reverberating circuits are correct, and if they are, they're only correct for short-term memory, and that means something else has to be going on in long-term memory. Or something else is happening completely. Damn it all, don't we have a thing here, a thing that fits our philosophical definition? We've got a change in matter, which is being read. The problem is that a lot of scientists don't study much philosophy, and a lot of philosophers don't study much science, because the fact that a thing may cover necessary conditions doesn't mean it covers sufficient conditions. Now, take this wonderful piece of paper that I put my A on here. By the definition of a physical change in matter that's read, this piece of paper can learn. Watch. Here, never mind there's an A on it, we don't care. Here's a physical change in matter. I fold it. That is the experience of this piece of paper. Has it learned? Watch. Every time I move the paper again, it folds at the same spot. The paper, because of its chemical consistency, is reading the physical change. Now, this pretty obviously is not memory. So we've got to start looking somewhere else than at the reverberating circuits. And of course the field did. By that point, as we move through the 20th century, we start knowing more and more about the chemistry of these wonderful neurotransmitters. That's the idea of, okay, fair enough. We don't really have memory in a big lump of brain here, this wrinkle or that wrinkle. And maybe we don't have it in the reverberating circuits, even though we might have that for short-term memory, but how the memory changes from that to something completely different, it really violates our principle of parsimony, looking for a simple explanation. So what else could be happening? Well, maybe memory is stored in the memory molecules. Maybe there's a memory molecule. And the obvious memory molecule would be a neurotransmitter. Now, lots of wonderful neurotransmitters. I have several models of them right here. Here, wonderful molecular model of dopamine. The black balls are carbon atoms. These are oxygens. There's other atoms here, okay? And this is the shape of the thing. And it's a kind of a great floppy molecule. And that's a bit of a problem, because suppose you want to store information on a molecule like this. Well, what's the best way to store information? Well, the most parsimonious way that anybody's ever found is the way memory is stored on a computer. It's stored as ones and zeros, this wonderful binomial system, ones and zeros. Okay? And the computer turns itself on and off very, very, very fast. That's a bit oversimplified, but you get the idea. And you can then give a talk like this on a wonderful video system here, because the computers stay very, very fast. Or you can look at whatever pictures you want to, and we won't judge you. Or you can write a paper. You do whatever you're doing with your wonderful computers here. It's all based on ones and zeros. But how would you code ones and zeros on this big floptious thing here? Here's another neurotransmitter, serotonin. Okay, look at the mighty floppy mess here. Okay, okay? Very difficult to figure out how you do your ones and zeros. But there are other neurotransmitters that ones and zeros would make a lot more sense on, and they're linear molecules. And one of the most important of these is this molecule right here, acetylcholine, or acetylcholine, depending what part of the country you're from. See, it's not some great flopsious ring compoundy thing. It's got kind of a nice linear way from this point here where it's blue is nitrogen, up to these two orange bits up here, this wonderful linkage here with the double O, the double bonded oxygen here. Okay, well, this would work. This would work because this nitrogen, right, is a lot of electronic activity going back and forth in this thing to these oxygens up here and this nitrogen back here, these wonderful negative elements from the periodic table. They're pulling back and forth. So you could see if you took this thing, right, you took a hell of a lot of them, and you might line them up somehow or other, kind of like old-fashioned printers type. Okay, maybe you get some kind of a printer device in your brain. You could line them all up and then put them in fields that would cause some of them to go so the electrons are relaxed, some of the electrons are coming down here toward what you could form a nice carboxylic ligature if you broke this in half. You could have that, and so you could have ones and zeros, either the molecule is on or it's off, ones or zeros, and I suppose you could have the whole thing spelling out hi mom or something. Now, if you look at a brain, of course, you find basically a big bunch of greasy, fatty electric head meat. There's no little square places there to field these out, but you could work that out later, I suppose. This seemed like an ideal memory molecule, and it especially seemed like one because there's a hell of a lot of it in neural systems that are connecting the hippocampus, well, connecting the limbic system anyway, to these upper parts of the cortex, where we think memory consolidation, or modern neuroscientists know that memory consolidation is continuing on far beyond the encoding functions of the hippocampus. Okay? So this seems like the molecule. And it was tested. Because you can't just keep making any kind of molecule in your body. If you did, you'd wind up being one kind of molecule only. You have to make them and break them down in a nice equilibrial pattern. Now, that means you need enzymes that break molecules down. The molecule that breaks down acetylcholine, or acetylcholine, depends on part of the country you're from, acetylcholine is acetylcholinesterase. Now, when you see ASE on the end of a chemical name, it means I break down whatever comes before me, okay, before the ASE, okay? So acetylcholinesterase breaks the molecule about here, and it basically forms an ester over here, and this group over here that becomes inactive. It breaks the molecule down. Now, in rats, first of all, rats have no legal rights. So in rats, you can go ahead and increase the amount of acetylcholinesterase compared to the amount of acetylcholine and break down the acetylcholine. Now, if it's the molecule of memory, the rat's ability to remember and to learn should go south. The rats should have poorer memory. Well, this was done, and it worked. When you destroy acetylcholine in mammals, and the rats are close enough to us compared to sea snails, that's a good bet we're on the right track here. Well, it turns out, son of a gun, and it was amazing. They discovered the molecule of memory. It was a huge deal. This was back when there was a little money around, so there was sweet and sour shrimp at the conferences. And amazing quantities of sweet and sour shrimp were washed down with champagne over the discovery. We had the molecule of memory. The only problem is there were competitors. By this time, the basics of protein synthesis worked out. DNA makes RNA, and RNA makes proteins out of amino acids. So that makes sense. And we won't go into the different kinds of RNA, but we can say this, that any given type of RNA is based on 4 base codons, 4 specific chemicals that make up RNA and its different configurations. Now, that RNA is a thing that actually carries a message, so to speak, from the DNA to when you actually, the messenger RNA is tRNA, you end up making the proteins, okay? It's carrying a message. And it has 4 atoms. Now, an interesting mathematical fact. Any map in the world, no matter how complex it is, can be drawn with only 4 colors. Yeah? Which actually, if you think about it, is just nothing at all. It's not pertinent in any way, but it sounds like it might be, huh? Okay, 4 is a good number. So, the dedicated scientists went ahead and, diminished the RNA factors in animals, and you'll never guess what happened. The damn animals couldn't remember as well. Okay. So now we've got a hell of a fight at the conferences between the friends of acetylcholine and the fans of RNA, and sweet and sour shrimp were not shared across the room. But suddenly, and enter, I want them to be holding a little flag with an AA on it, the fans of amino acids, specifically the pals of protein. Now look, amino acids, there's a hell of a lot of them. I think there's over 600 of the damn things that are known to chemists. But in terms of important amino acids that act in living systems, there's really only 20 of them. Now, but think about that. How many letters are there in the English alphabet? 26. How many important naturally occurring amino acids in life? 20. 20, 26. If you have a certain kind of mind, that means something. Well, you'll never guess what they did. They reduced protein synthesis in the rat, and the damn rats lost their memory. So now we've got a three-way split at the conferences. You've got the fans of acetylcholine over here, and the friends of RNA over here, and the pals of protein over here, each with its own bowl of sweet and sour shrimp and their own champagne, and they were not sharing. Okay, so what in God's name was happening here? We didn't get it. Psychologists and neurobiologists, we didn't get it. We just didn't understand. But the English professor and novelist John Gardner understood. He did. One of his novels, he has a character say, take crabs, for example. We poke them with a stick, and they behave as if they've been poked with a stick. In other words, when you reduce a rat's protein synthesis by about 3/4, you know what you get? You don't have a rat whose memory has been damaged. You have a damn starving rat, okay? His whole system's falling apart. He doesn't give a damn about his memory test. All he can do is crawl along going, dear God, Velveeta, clunk, okay? He's not doing well. And the same is true if you start screwing around with living creatures' neurotransmitter systems. RNA is the basis of protein synthesis, and since our muscles are made out of protein, a lot of us is made out of protein, you basically just... so to speak, put, a bullet through your rat and expect him to do well on his memory test. A damaged animal is not an animal plus the damage with reference to whatever we're studying. A damaged animal is an animal with that damage going through its entire holistic system. This simply wasn't somehow, it was missed. And funding for the study of the memory molecule dried up. Now that's it. Maybe it shouldn't have dried up quite that quickly. Because of something almost nobody talks about anymore, it's too embarrassing. The disquieting mystery of worms. There's a kind of creature called a flatworm. It's almost microscopic. If you have really bad vision, so you take your glasses off and your eyes are like magnifying glasses like mine, you can just make them out. The tiny, tiny, tiny animals. They're flatworms. Planaria, the platyhelminthes, which means flatworms, okay? And they have a very simple nervous system, but they are an animal that has a nervous system. They're probably the simplest creature, this could be debated, but the simplest creature that you can routinely manage to do a bit of conditioning with, okay? Well, these worms are already afraid of light. They're photophobic and they're negatively phototropic, but they can be conditioned with a few little wormy shocks to be even more afraid of light. Good so far? Well, flatworms were taught to be even more afraid of light, and then they were ground up and fed as delicious wormy shakes to their brother worms, all right? Well, in some experiments, it turned out that the worms who'd eaten their brothers in a cannibalistic frenzy were now more than normally afraid of light. It looked like they'd eaten the memory. Well, labs all over the world were trading in their rats for worms and microscopes, having a wonderful time, and it turned out that most labs couldn't get the effect. A few labs did get the effect, most could not get the effect. An American psychologist named Kandel didn't help this any. He founded a journal based on this. One of the problems in this field is very few people have much of a sense of humor, and McConnell named his damn journal the Worm Runner's Digest. Not only did he put serious studies in there, but he had a few joke articles too. People just did not buy into this, okay? And so the whole worm thing started to become science by democracy, I guess. Since most labs couldn't do it, it started to die out. Well, then a couple of labs reported it in rat, okay? Now, they didn't feed the rats the whole other rat. They took out their little brain, ground it up in a rat-o-matic blender or something, and fed the brain in a delicious, ratty, brainy rodent shake to their brothers' rats, and damned if in a few labs the things suddenly seemed to have the memory that had come with the brain. Well, again, a few labs did it. Most labs couldn't do it. Science by democracy was laughed at. The last time I saw anybody trying this, there was some lab down, I think it was Mexico City, that tried this with cat. They reported in one cat in the 1980s. I didn't even think there was any follow-up on that. Because the fact is, most labs simply couldn't do it. Now, that said, science by democracy has never worked. There's something funny here. But if any students are listening to this or watching this, what I can say to you is, please don't try to take this up as your dissertation. Because for one thing, you'll be laughed out of the market. For another thing, we now know, thanks to the neuroscientists, been tracing the consolidation of memory from the encoding, the initial consolidation of the hippocampus, right into the cortex. There's a tremendous amount of molecular interaction here. It's very probable in my mind, and feel free to disagree, I could be wrong, but maybe you don't have the molecule of memory there. Maybe you have some molecule that is involved in the production of memory. So why did it happen in some labs and not in others? My best guess, and I've never heard anybody else say this, so you heard it here first, folks, on The World of the Mind. Whatever the molecule is, sometimes it can get through a digestive system, but most of the times it can't. But if you find it, I think you're going to find some intermediate step that happens here and there. It may already have been discovered without anybody knowing it. It's really not worth following, and so to my mind, anyway, I could be wrong, but not at a high level. Not, oh my gosh, we've got the molecule of memory. Let's teach the molecules some physics and inject it into people. We'll make physicists. That's not going to happen. Okay. So funding for the molecules dried up, especially in the wake of poor McConnell and his quite amusing Worm Runner's Digest. So what the hell do you do next? My God, we're starting to exhaust physical reality. We've exhausted the physical structures of the brain, these big wrinkles, and it looks like we've exhausted the circuits of the neurons. Now we've exhausted the molecule. Well, where do we look next? The atom? Well, that could be. You could have some weird deal that some specific atom, maybe in a particular molecule, we don't know. Let's say maybe this carbon, third carbon from the left here. Then you get to a higher orbital, and that's 1, and you get to a lower orbital at 0. That's possible. But if that was happening, electroconvulsive shock and epileptic seizures would wipe out the memory. So it can't be in the electrons of the atom. Could it be in the nucleus of the atom? No. Because that would involve nuclear energies. Every time you remembered something, your hometown would disappear under a mushroom cloud. Okay, that's not exactly accurate, okay? But the point is, sorry, bad shrink, no donut. But nuclear energies are of a type, the strong nuclear forces, of a type that you simply could not be manipulating this in the service of memory. It doesn't work. So what's left? Well, what's left gets into the theoretical boondocks very rapidly. The only thing left is quantum mechanics. Now, the great physicist Richard Feynman once said, and I think he's probably right, that nobody understands quantum mechanics. There's a hell of a lot of it that's not only counterintuitive, but seems to contradict other physics in relativity and so forth. It's quite a mess, okay? And the fact is, quantum mechanics, well, it's so bizarre, it doesn't even make sense, and yet it's the most tested theory in human history, according to the history of science people. Every time you nuke a Pop-Tart in the microwave, you're using quantum mechanics, so you can't really ignore this theory. And one of the consequences of quantum mechanics, the behavior of the particles that are even smaller than the particles of the nucleus, one of the factors here is J.S. Bell's molecular bifurcation theorem. This is a serious concept because Bell's bifurcation theorem suggests that if two particles originate in a given atom, they will record changes in each of the term is usually called spin, but that isn't really correct. They record changes in each other's physical characteristics anywhere in the universe. In other words, distance doesn't matter, origin does. Now, this is a very bizarre idea. If you want to kiss somebody, you have to be within lip range. You can't kiss them if you're in Boulder and they're in Schenectady. Distance matters, doesn't it? If you want to whack somebody in the head with a club, they have to be within baseball bat range, okay? Distance matters, but what Bell suggests is that at the quantum level, that may not be the case. Distance may not be the point. The point may be origin. And there have been experiments done, obviously not across the universe, but over distances of a few meters, where it seems to be the case that what you seem to record these changes in spin without reference to distance, or changes in physical characteristics without reference to distance. Well, if you do, that means a particle 1 can record particle 2, and that gives you your 1s and 0s. Now, this is possible. Now, practical experimental psychologists, tend to doubt this quite a bit, in part because how the hell would you possibly integrate that into the molecular and cellular structures of the brain? It just seems too far down the material realm. But let's suppose it is true. Let's suppose it's possibly true. We have another problem here, and that problem is the Heisenberg uncertainty principle. Now, all sorts of rubbish has been written about the Heisenberg uncertainty principle, and lots of it turns out to be just complete misapplication, okay, in many of the sciences, including psychology, okay. Basic idea is, oh, well, if you're in your field situation, you contaminate it, so you can't be sure what's happening, haha. You know, in other words, if the monkeys can see you, the monkeys will behave differently. And frankly, having been in that exact situation, when the monkeys did see me, they did behave differently, okay, so it did, and they was, okay. But, that isn't really the principle. The principle is this, that you cannot really record specific physical characteristics of the same particle and get them both right. The classic vector is either velocity versus the positioning. You cannot know them both. And to make experimental sense out of the Bell molecular bifurcation theorem as a basis of memory, you would have to. So we've got this problem here, as you can see on this paper I'm putting up here right now. Okay, and by the way, I did present this once at a conference years ago. I'm not sure everybody was too amused, but I do make these graphics myself, and tremendous expense is spared whenever I do so. But basically what you're looking at here is the progression of our understanding of the engram. Because at first you can have the idea that the brain is telling you, your big chunk of the brain has don't eat bugs on it, okay? Well, that didn't work. So then you go to the reverberating circuits with giving you little messages. Well, that didn't work necessarily because electric shocks would knock them out. So then you go to the molecule running around telling you stuff. And obviously we wouldn't, the diagram's a bit facetious here. We're not really suggesting that the molecule is carrying a little sign. But the molecule's now out. Well, the atom's out for the reasons we just got to. And so now we're down to the quantum level. And J.S. Bell says maybe the molecular bifurcation theorem could somehow be integrated with the brain. But Heisenberg says, sorry guys, sorry people, you'll never know. That's where we are right now. We don't know. Now, the neuroscience people are doing some amazing work now. They're starting to look into what is happening in the cortex as consolidation goes on from the hippocampus, which we've known for a very long time, right up there into the higher cortical centers, the dorsolateral prefrontal cortex, very mysterious structure, does all sorts of interesting things. There's a lot of modern new work going on. It's very important work. But what it's still getting at, really, is what experimental psychology gets at. Not what a memory is, but how it works. And what we've talked about today shows us something of great importance, I think, and it is aporetic. It's like Socrates talking to Meno saying, well, that's not it either, okay? Not that I'm comparing myself to Socrates, it might be a bit more like Meno, but the point is, we can't get there from here. We have exhausted physical reality. So somehow memory is operating as some integration of these systems. It has to, because unless memory is part of the immortal soul and it's happening in a supernatural realm, and virtually nobody thinks that, at least nobody you'd want to talk to for very long, somehow, someplace in the interaction of these systems, memory must exist. And not only is it a real thing, with real consequences. You want your architect to remember architecture. You don't really care if he or she remembers dermatology, okay? If you're itching, you don't need an architect. You need somebody with a memory of dermatology, right? Okay, so memory is awfully damn important. But it's not like this pencil. It can suddenly turn into an iguana or a pipe wrench or a hyena, okay? So what we're stuck with then is trying to understand the nature of memory at the functional level, both psychologically and neurobiologically. That's where we are right now. And we know a bit about this. We may be able to explain today focal retrograde amnesia. We can also, as you see in videos here, we can also make this very useful for the student in improving his or her performance so that he or she can become an architect or a dermatologist. We can work a lot with memory, even though we're in this very paradoxical situation. We don't know exactly what it is. But we'll continue with that in future videos here. I hope you found this interesting and useful, and I hope you'll subscribe if you feel like it. I'm Dr. Matthew Sharps. This is the World of the Mind.